SDS PAGE electrophoresis is a variant of the polyacrylamide gel electrophoresis. It uses sodium dodecyl molecules, SDS, to help identify and isolate protein molecules. SDS PAGE electrophoresis was developed by Ulrich K. Lamelli and allows a good separation of proteins with molecular masses between 5 and 250 kilodaltons. This method works in a similar way as the polyacrylamide gel electrophoresis, which separates charged molecules in a mixture by their molecular masses in an electric field. There are three things that determine the movements of any charged species through an electric field: its net charge, its molecular radius, and the magnitude of the amplified field. The problem with proteins is as they have different 3D structures, the migration speed will be dependent of the structure and their charge. So to separate proteins only based on their molecular weight, the tertiary structure should be reduced into a linear molecule. That's when SDS comes into action. SDS is a detergent present in the buffer that brings unfolded proteins into linear ones. Also, cut the proteins with negative charge by masking the charge on the R groups. This helps to not having a migration based on size, but only on molecular weight. In this practice, yet, the objective protein is GFP, which is overexpressed in E. coli. This protein is conformed of about 238 amino acids and have an average size of 26.9 kilodaltons. As commented before, this protein was first isolated from the jellyfish Aqueuroa victoria, and in molecular biology, this protein is used as a reporter of expression and sometimes as a biosensor. The materials used for this practice are amphisilin white and amphisilin arabinosine green plates from the Transformation Lab, DTT, which reduces disulfide bonds, Precision Plus Kaleidoscope, which is a mixture of 10 multicolor recombinant proteins and serves as a ladder, BioSafe R Comasi stain, which stains the proteins we're looking for, Tris glycine SDS buffer, which, as mentioned before, disrupt the tertiary structure in proteins and neutralize charge. The lamelli buffer, which also helps to break down the disulfide bonds because of it has beta-mercaptoethanol, DTT, bromophenol blue, that it's an indicator dye, and SDS detergent. The ready gel R, 4 to 20%, which is a matrix of the electrophoresis, pipettes, screw cap microtest tubes, foam microtest tube holders, transillumination, water bath and thermometer, dyeing frames with clamps, cellophane, micropipettes, permanent markers, distilled water, and minipotent tetracell, and a power supply.
Two microtubes were labeled as follow: G-H, which means green and no heat, and G+H, which means green and heat.
300 microliters of Laemmle sample buffer were added to each of the microtubes.
By using the inoculation loop, 1/4 of the total transformed colonies with GFP were scrapped and transferred to the plus heat tube. In the other tube, all the transformed colonies from another petri dish were scrapped and added.
Both microtubes were mixed by spinning the loop with the thumb and forefinger. Visible clumps were still present, so we proceeded to pipette both solutions until clumps were totally dissolved.
The plus heat tube was submitted to a water bath at 95 degrees Celsius for 10 minutes and then cooled at room temperature.
For the SDS page, a 4 to 20% 10-well rated gel for electrophoresis in a mini protein tetracell was used.
Samples were loaded with the following order: the 2 plus heat tubes first, and then 2 not heat tubes after using a micropipette at 15 microliters.
The polyacrylamide gels were electrophorosed for 30 minutes at 200 volts in 1 times TGS buffer.
After the time passed, the gels were removed from the vertical chamber and introduced in a transilluminator.
The bands from the samples that were not treated with heat showed green fluorescence while the others did not.
After visualizing the gel in the transilluminator, they were rinsed in the water in a rocker all night.
Finally, for the gel staining, the previously used water was replaced with 50 milliliters of BioSafe Comasi stain. After staining, gels were rinsed again with water.
As reported by Shapiro, polyacrylamide gels are widely used for protein separation. They are formed by polymerization of acrylamide and a cross-linking reagent. The oxygen radicals are generated from the catalyst ammonium persulfate when it reacts with the second catalyst, timide. The size of the pores can be adjusted by the same principle as in the gels for the previous sessions, increasing or decreasing the concentration of the component in the gels, in this case acrylamide or bisacrylamide. The difference in working with proteins rather than DNA or RNA molecules is that they have a diverse structure. Proteins have hydrophobic and hydrophilic regions, and when they are folded, assume many different geometries, and their surface are mosaics with respect to the distribution of the R groups with different chemistries. These complex structures are the reason that the molecular weights cannot be determined in an electric field. In a SDS page, proteins are denatured by increasing the temperature while being in an anionic detergent solution, SDS, and beta-mercaptoethanol. This action breaks the covalent bonds and the SDS permeates the protein to make it negatively charged. For this laboratory practice, Shimomura and Johnson protocol for GFP protein extraction and purification was used, with modifications made by PhD Victor Rodriguez. The results of the SDS page after UV light exposure show the 4 wells used by our team. The first 2 wells showed no visible band, and the last 2 showed a green fluorescent band in both of them. According to Ishii, the denaturing temperature for GFP protein varies between 80 degrees and 95 degrees. The first 2 samples were submitted to a water bath at 95 degrees. This explains why the wells for these samples show no band under UV light. They were completely denatured. Even though a partial denaturalization of the protein was required to perform the SDS page, the region of the protein responsible for its color is within the tertiary viral structure of the protein. This region consists of 3 adjacent amino acids on the primary amino acid backbone, and the denaturants cannot affect it. The results shown by the SDS page were the expected ones. As Byrat states, the visualization of the GFP fluorescence was apparent only on the unheated samples. Both bands shown in the electrophoresis were well defined. Meanwhile, the bands of the samples that heat was applied did not appear as expected. Both bands that were not heated had a similar size as the first fluorescent band shown in the kaleidoscope ladder, which had a size of 70 kilodaltons. Hink and collaborators on their page named Structural Dynamics of Green Fluorescent Protein Alone and Fused with a Single-Chain FB Protein presented that the denatured GFP has a molecular mass of 27 kilodalton, a weight very close to the obtained in this practical, as the bands obtained were between 25 and 30 kilodaltons.
This table shows the molecular weight and the distance on the gel after the SDA's page of the following proteins compressed in the kaleidoscope ladder. The following values represent the distance of the fluorescent and non-fluorescent green proteins in millimeters that travel across the polyacrylamide gel. This graph shows a standard curve and estimation of the different molecular weights for the different GFP conformations. This curve was generated by the plotting of the logarithm base 10 of the molecular weights versus the distance migrated, in millimeters.
The SDS page is a technique that can be used to identify a certain protein after it has been extracted from an organism. The identification of these proteins is based on the different molecular weights for each protein. During this practice, we'll learn that when you are looking for a specific protein, it can be shown in the electrophoresis gel as different conformations depending if they were denatured or not. This shows the behavior that the proteins can have within an organism in response of different environmental factors, as in this case, temperature.
The SDS page electrophoresis is a really important method used in genetic engineering because it helps to identify and isolate proteins. This, in the medical and pharmaceutical industry, helps to determine and potentially treat diseases and conditions, which without the proper identification of proteins will be nearly impossible. Also, protein identification helps to identify the secondary metabolites of many metabolic routes, which can potentially be used in the industry.